To replicate the DNA, a variety of enzymes are needed. And enzymes are proteins that help to speed up specific chemical reactions. So for example, replicating the DNA is a series of chemical reactions. So this particular portion from your book shows different enzymes that are involved in this process. And one of the characteristics you can see of enzymes, and this isn't always the case, but it is in this example, is that enzymes end with an ASE. So the enzymes we're going to look at are helicase, gyrase, primase, DNA polymerase, ligase, and telomerase. Now, we said in order for the DNA to replicate, the DNA double helix has to be opened up or denatured. The enzyme responsible for that is helicase. Now, when you're opening up this DNA double helix, there can be twisting that occurs, and that twisting could ultimately form, quote, knots in the DNA. So another enzyme prevents the building up of those knots, and that's called gyrase. To start DNA replication, DNA polymerase has to have a short piece of nucleic acid already there. It can't start from scratch. So the enzyme responsible for laying down the short piece of nucleic acid to allow DNA polymerase to actually start DNA duplication is called primase. Now it's interesting to note that this short piece of short piece of nucleic acid that primase lays down is actually RNA, not DNA originally. The RNA will be removed when the different DNA fragments get joined together as a result of replication. The enzyme we said that actually replicates the DNA is called DNA polymerase. Eukaryotes have 3 different forms of DNA polymerase. The one responsible for transcribing genes or making products from genes that will ultimately be used to make proteins is called DNA polymerase II. So what DNA polymerase does, we said, is it starts off of this short primer and it replicates the DNA on the basis of complementarity. So new DNTPs will be hooked together by a phosphodiester bond to build the new complementary strand of DNA, again, complementary to the template. Now, if you take another class in genetics, you'll go into a little more detail about the process of replication. But the idea is DNA replicates in multiple intervals at the same time. And when all of these intervals are done, you have to join the intervals together to create the finished product. So the enzyme responsible for joining those different pieces of DNA together to create the final product is called ligase. Or you could think of to ligate, which means to hook together. So ligase is the enzyme that does that. We've talked earlier about the telomeres, and we said the telomeres are ends of chromosomes, and they have to replicate in a very special way. The enzyme responsible for doing that is called telomerase. Now, in our cells, telomerase is typically not functional. In other words, it's important for the lengths of the chromosomes to shorten to provide a signal to the cell that it's time for that particular cell to stop replicating. However, in cancerous cells, cells with uncontrolled cell division, telomerase is typically active in those cells. So again, we see a list of enzymes that are involved in the process of replicating DNA. So you should start to get a sense of how complex this particular process is.